Hello, everyone. So it's again my great pleasure to introduce our next speaker. I was just commenting with Michael that both Michael and Susanna gave plenary talks in the last International Botanical Meeting in Shenzhen, China. I've been recommended to summarize Michael's career in 1 minute, but it's really very difficult, as you will see soon. His research career has spanned many different institutions: San Diego State University, Arizona University, Harvard, Yale, and now back to Arizona. And he also maintains research professorship in Yale, and has been director of Harvard University Herbaria, director of Peabody Museum, Natural History in Harvard, Evelyn Hutchison Professor. He's been director of the Yale Institute for Biopherical Studies, and many professional recognitions, fellow of the American Association of Atmospheric Science, is a member of the National Academy of Science, has been president of the Society of Systematic Biologists. If I go through his career, the numbers are mind-blowing, around 300 papers, and very important, for me at least, is that he has supervised many doctoral and graduate students who has become later prominent figures in evolutionary biology and botany. I went through the list and I could recognize all those names, and many of them are actually here. But I want to maybe give a bit of hints on his research contributions. And again, there are many. Michael has worked on the tree of life of green plants, has been a very important figure in linking evolution and ecology. I can remember his main sentence, plants can move, but it's very difficult for them to evolve, to develop new climatic tolerances. He has gone to the idea of innovations, the confluence of multiple key innovations in the tree of life of green plants. And he has been a prominent figure in the defense of model clades, the importance to understand general patterns through in-depth studies of a plant clade. Understanding physiology, ecology, evolution, genetic architecture is the only way we can find mechanisms and to understand the patterns. And that has inspired my career, actually, Michael. And just to finalize, I will say that Michael, I think, still plays banjo, because I've been in several meetings with him, and he is one of a prominent dancer. So without further ado, I want to give the microphone to Michael. Thank you.
Hello, everyone. It's wonderful to be here. Buenos dias. And Isabel, thank you so much for that introduction. That was fantastic. Anyway, I'm delighted to be here. I'm thankful to the organizers for inviting me, and it's a wonderful meeting so far. It's just going to be tremendous to be here with all of you in Madrid, old friends, new friends. It's just great. So I'm going to get started talking about plant geography, and the talk that I'm going to give has 3 little parts to it. The first one is somewhat short, about the latitudinal diversity gradient. Then I'm going to talk about northern hemisphere biogeography through most of the middle part of the talk, and then I'm going to end with a commentary, I guess, on how we can do a better job of assessing ancestral areas and ancestral biomes. So let me see if I can operate the machinery here. Right, so just as a bit of background, in my view, broadly speaking, biogeography falls into 2 main categories. One is what's called ecological biogeography, focused on diversity in relation to things like latitude, elevation, and so forth, spanning from island biogeography to modern macroecology, and traces back basically to Humboldt. On the other hand, there's historical biogeography that's focused on areas of endemism, disjunctions, where did clades originate, how did they move around, how was that movement affecting their diversification, and so forth. That thread traces back to Alfred Russel Wallace and, of course, Charles Darwin. Phylogenies now play a major role in both of these areas, and this has happened over the last couple of decades, and has served actually to kind of reunite these things and sort of bring them together in a way that phylogenies have tended to do, just as they have done with population genetics and macroevolution, for example. So phylogenies are playing a major role in all of this, and I'll try to illustrate that as we go. So the first vignette is about ecological biogeography, and specifically talking about the latitudinal species diversity gradient. And I think this is a familiar idea to everybody in the room, and that is that biodiversity isn't sort of randomly distributed. It looks like there's more species in tropical latitudes than there are as you move towards either pole. So I think that's a pattern that's well known in plants, as you can see on this kind of map, but it's well known in many other groups of organisms as well. And it's taken up a lot of time in the literature, people thinking, why is this the case that we have more species in the tropics? There are two major sets of explanations. Each one of these has sort of subsets, but two major sets. One is that diversification rates are greater in the tropics for whatever reason. And the other is that there's been a longer time to diversify in the tropics than in the temperate zone. And that's illustrated in those two graphs there from a very influential paper by Mittelbach et al. That sort of outlined the variety of hypotheses and tried to evaluate them. So in 2004, 20 years ago, I published a paper with John Wiens in Trends in Ecology and Evolution where we talked about this problem from a sort of phylogenetic standpoint and offered a fairly straightforward historical explanation. And we gave it a name. We called it the tropical niche conservatism hypothesis, and that's been pretty widely cited by now. But I want to make clear that we just kind of gave it a name, but it was already kind of an existing idea in the literature, going back to Fisher, going back to Farrell. The paper by Latham and Rickliffs that I've sort of highlighted in red was particularly influential in my own thinking about this problem. But anyway, we brought it together and sort of offered up this hypothesis. And the idea is you have more species than tropics. Why is that? Well, maybe it's because groups started out diversifying in the tropics and only later moved out into the temperate zones, into colder climates. And so they've had more time to diversify in the tropics than they have in the temperate zone. That's a very straightforward historical explanation. And notice that it doesn't have anything to say about things like diversification rate. I never mentioned that, right? So there are basically three elements of this hypothesis, and I want to lay them out so that we can see how they can be tested. One is that the idea that more groups originated in the tropics. Many clades originated in the tropics because the tropics were much more extensive before 30 or 40 million years ago when climate started to cool down. So there was a broader swath of tropical climate, and many groups originated in that area. The second element is that clades that originated in the tropics dispersed to the temperate zone later, sort of more recently, they've moved out into the temperate zone. So they've had less time to diversify in the temperate zone than they have in the tropics. And the third element of this, the way we laid it out, was that there's a bit of an impediment to moving into temperate climates, that there's some sort of inability of some organisms to adapt to those freezing climates, whereas other groups are able to adapt more readily to those climates. So those are the elements of this hypothesis. But on that last note about conservatism, I want to make sure to mention that this is a much more complicated problem than just thinking about the accessibility of the relevant adaptations, like adaptation to freezing climates, because there are other things that will determine or help to determine whether things make a biome shift from, say, tropical to temperate or vice versa. And that is geographic adjacency and geographic opportunity to connect between areas, and biotic interactions with organisms in new areas that are about to be occupied. So it's not just the accessibility of relevant adaptations, and I just want to put that on the table to begin with. But anyway, these elements can be tested phylogenetically, and John Wiens and I recently did a project, which we've written up now, but it's not published, where we said, let's go through the literature and see which papers have actually tested these different propositions, and where do they come down? I mean, what does the evidence suggest at this stage? What can we say in general about this, given the literature? So we can test, did clades with higher tropical species richness originate in the tropics? That's one element of the hypothesis. Do clades with high tropical species richness have higher phylogenetic diversity in the tropics? Because perhaps they retained older lineages there. Is there evidence for climatic niche conservatism, especially with respect to freezing? So we can go through papers and evaluate whether they tested these things. As I said, technically the tropical conservatism hypothesis is neutral with respect to diversification rate, but for the sake of completeness, we can also look at papers and say, did they test that one? And what do they have to say about diversification rates in the tropics versus the temperate zone? So we did that, we did a kind of meta-analysis of 219 papers that were selected from the literature using Google searches. And 89 of those are animals, 127 are plants. I did all the plant ones, John looked at all the animal ones. But we put them together into a database that looked like this. So there's the study, the group of organisms, the age of the clade, the spatial scale, what kind of richness pattern it is. Did they do ancestral reconstructions? Are those consistent with the tropical conservative hypothesis or not? Did they do phylogenetic diversity measures? What do those say? Did they do conservatism tests? What do those say? So we went through the literature and scored them. I'll give you an idea that it's a very interesting literature, by the way, to step back and read through all of these papers. And some of it's highly satisfying and some of it's kind of not so satisfying, but I won't go into that aspect of it. But I'll just give you a kind of a quick slideshow of a couple of these studies. So this is one by Kirchhoff et al. In 2014. So we were studying papers between 2006 and 2023. So these are New World woody angiosperms. They had a tropicality index, which they mapped on a tree of angiosperms. And as you can see, the red are more tropical, the blue are more temperate. And this is very consistent overall with the idea that angiosperms originated and started to diversify and had more time to diversify in tropical climates and moved out more recently into temperate climates where they've had less time to diversify. So that's just an example of that sort of study. There were a whole bunch of studies, especially in plants, that dealt with measures of phylogenetic diversity. And this is just an example of one of those. This is angiosperm trees from North America. And the idea is that so you take, pixel by pixel, you can measure the phylogenetic diversity and use various measures like this PSV and NRI, different phylogenetic diversity methods. And basically what this shows is that species are generally more phylogenetically clustered and clades are younger as you get up into the more northern latitudes. So this is one that's consistent, again, with the tropical conservatism hypothesis. Here's one that deals with diversification rate. This was a study by Sun et al. On roseate angiosperms with a big roseate phylogeny there. And they showed quite nicely, I think, that diversification rates were actually higher in the clades that were outside of the tropics. So that's, inconsistent with diversification rate hypothesis. Anyway, that's what the literature looks like. When you summarize it, I just have one little summary graph with some statistics here. This is what it looks like, and I'll just summarize this very quickly. So overall of this data set, what we can say is, yes, there's greater species diversity in the ancestral area in the tropics. So there's support for that element of the tropical conservatism hypothesis. Yes, there's greater phylogenetic diversity overall in the ancestral area than in the more derivative areas. So that supports the TCH. And yes, there's evidence for tropical niche conservatism, and especially with respect to freezing. But diversification rate doesn't fare so well. And in fact, in many of the studies that we examined, the diversification rates weren't higher in the tropics, they were higher outside of the tropics. So those are the basic results here. So those are the conclusions. The predictions of the TCH have seemed to hold up pretty strongly in the studies that actually did the relevant tests. Diversification rate hypothesis, on the other hand, has not fared as well, like I said. Clades are often not diversifying faster in the tropics, more so outside of the tropics. As I was evaluating this literature myself, it seemed that there's a whole bunch of issues that need to be addressed. One is I came to the conclusion that phylogenetic diversity measures are actually very indirect and often hard to interpret in this regard, and especially when people use things like mean family age as the thing that's being measured. These are really hard to interpret, and there's alternative explanations for all of these things. Conservatism tests, likewise, I think are very indirect and don't always address, this adaptation to freezing aspect that is so critical to this hypothesis. And then finally, very few studies actually did all the possible tests that you could do, all the ones that we were looking for. In fact, I think only one actually did all of them. But in particular, there were very little attention, and I recommend this to any of you who want to do it, to testing colonization time directly as a factor. So in other words, how long have you stayed in a particular climate, and how does that relate to your diversity? And actually, among all of the plant studies, there was only one study that actually tested that. So we're in need of a lot, further tests. But this is where we stand, sort of 20 years into this. Okay, so that's the first little part. Now the second part's about historical biogeography. And what I want to talk about here is mostly the northern hemisphere biogeographic problem. But I want to give a little bit of historical background at this point about developments in this area. So I'm going to start with Philip Darlington, who I suspect many people in the audience will not have ever heard of. He's a zoologist who was a professor at Harvard, and he was a very important biogeography person in the old days, in the middle 1900s. And he published a book about the southern hemisphere biogeography. And he was roundly criticized by people at that time and later by others for basically acting as though plants and animals were distributing themselves over a more or less static modern landscape, even though we knew at that point about things like continental drift, right? But he stood by it, and so did George Gaylord Simpson, and both of them were criticized in the same way for interpreting biogeography on a static, more or less modern landscape. Take a look at that diagram, though. That's basically the movement he inferred for this was before phylogenies. The movement he inferred for 3 different major groups of amphibians. And it basically shows the continents sort of separated from one another and things originating in the old world tropics moving into the northern hemisphere and around into the new world and so forth. And I want you to sort of just get that in your brain, because I'm going to return to that slide a little bit later. But that's a sort of starting point. At around that same time, Willy Hennig published about phylogenetic systematics, and in that book, he for the first time was putting phylogenetic trees onto maps, geographic maps, and he came up with something he called the progression rule, which is a sort of, in a sense, it wasn't really a method, but it was a way to sort of think about ancestral areas and movements based on putting the phylogeny on the tree. A friend of Willy Hennig's, who also studied flies, was Lars Brundin, and Lars Brundin said, well, let's just not look at single groups of flies or something, let's look at multiple lineages and see whether we see congruent patterns in them. That was a very interesting step to take, and in fact, he actually did it with chironomid midges, which is a group of Diptera. He looked at multiple clades of these midges around the southern hemisphere and found good concordance between them, and he basically ended up concluding that, as I said here, there's not the slightest evidence of chance dispersal over wide stretches of ocean. He interpreted all of this congruence of these phylogenies in terms of the breakup of the southern continents, basically. He didn't have any information on the absolute timing of the midges, which is interesting, but that's how he interpreted it. Well, that became basically the starting point, if you will, for what became vicariance biogeography, the vicariance approach to biogeography. If you haven't seen this paper by Nelson and Platnick in 1980, if you're interested in history of any of this, I recommend that. It's a short piece that really outlines the basic ideas. And I'll just read it briefly. It says, biologists must produce detailed studies of the interrelationships of endemic taxa of various groups of organisms, summarize those in species cladograms, but then compare species cladograms of various organisms in search for general patterns of geographic relationship that can be summarized in biological area cladograms. So the idea is you look at this group, you look at another group, you look at another group, you see whether they have similar patterns. And you sort of build that into a general biological area cladogram, which then could be compared to a phylogeny, if you will, of the continents to see if it matches the continental breakups and so forth. That was the idea. Now, it was a good idea. I mean, that was a very laudable idea. Like, let's look for general patterns. And in fact, we've gotten away from that, and I'm trying to get back to it, as a matter of fact. But so I wanted to introduce that. Now, I should say, after the 1980s, as we got into the 1990s, there was a shift in the literature back to the question, the sort of Darwin, Russell, Wallace type questions of ancestral areas and how things had moved around. And the beginning of that was really a botanist, Corey Bremer from Sweden, who published a paper on the origins of the Asteraceae. And he reasoned, based on this phylogeny at the base of the Asteraceae, that Asteraceae most likely originated in South America. That touched off a series of papers, or a controversy, if you will, with another Swedish guy, Fred Ronquist. And Frederick then, in relation to Bremer's paper, ended up developing something called DIVA, which is dispersal vicariance analysis, which is a parsimony-based method for making inferences about ancestral areas. And that became very popular in the 1990s and through the early 2000s. So this is not looking for general patterns or anything. This is looking on individual phylogenies and trying to reconstruct where things have been in the past and how they've moved. And this has been very profitable and very big in the literature. Starting in 2005, we moved from a parsimony-based sort of approach to this to a more statistical model-based approaches to reconstructing ancestral areas and everything. And in particular, I think that was touched off by a paper by Rick Ree and others. And as it says here, this approach incorporates information on the timing of both lineage divergence and the availability of connections between areas. And so I think it was quite a nice advance. Now, there have been many further advances along those lines on what's called the dispersal extinction cladogenesis model. And in the hands of Stephen Smith and especially Isabel, who's sitting right there, has done a lot with this. Nick Matsky, Michael Landis, a whole team of people have worked on developing better models for inferring ancestral areas, and then also developing software like Lagrange and biogeobears and other such things that you've heard about. Okay, so now with that background, let me get into this Northern Hemisphere biogeography. So there are many studies of individual clades, and I want to say, is there a general biological area cladogram that we can draw for Northern Hemisphere plants? And if the answer is yes, then what factors are responsible? And if the answer is no, then why not? So that's where I'm going with this segment. Now, my own interest, a little personal note here, my own interest in this goes back to a particular day, actually, in 1972. So that was, 52 years ago or something. And that's me at the age of 19. And I went on a field trip with a guy, my professor, his name was Jay Harmon. And he took me, along with several grad students, down to the Smoky Mountains. And we spent a couple of weeks collecting plants. And this was my first actual field trip anywhere. And I was just completely thrilled. And that trip turned me into a botanist almost instantly. And so this is a picture of us standing around a Liriodendron tulipifera tree, what we call a tulip tree, a giant tulip tree in the southern Appalachians. What could be cooler than that? And I was just thrilled by that and by all the plants. And I said, where did all these amazing plants come from? And I turned to my professor and he said, I don't know. I don't know where they came from, but I do know that they have relatives in East Asia. And I said this about Liriodendron, and I said it, we turned to rhododendron, and he said the same thing. And sure enough, it's really true. Liriodendron tulipifera has its closest relative in China. And it was on that same field trip that I was introduced to Viburnum lanthanoides, that lives at high elevations in the southern Appalachians. And I realized after some while that that's another example of something that has its closest relative in Eastern Asia, Viburnum furcatum in Japan. So I was hooked on this problem from a fairly early age and have continued to be fascinated by it. There are many, many of these East Asia, Eastern North America disjunctions. Pieris is one that had 5 species in East Asia, 3 species in North America. Liquid ambar shows a pattern like that, but it also includes a species in Europe. And then there are plants like Platanus, in this case, that are present in Eastern North America, Eastern Asia, Europe, and also in Western North America. And those turn out to be 4 sort of general areas of endemism around the Northern Hemisphere that we can talk about. And there are many plants that display distributions that look like this. And if you add in the fossil information, it gets even more interesting. So here's a group, Caria, that is distributed today with extant organisms in Eastern Asia and Eastern North America. But if you add in the fossils, you can see they were present abundantly in Europe and also present in Western North America. And so we have very good fossils from the late Eocene of Oregon, for example, even though they're not present there now. So altogether, what this amounted to was the idea that there must have been some widespread forest type around the Northern Hemisphere where plants were widespread around the Northern Hemisphere. This is a drawing of what might be called the mixed mesophytic forest in the mid-Miocene, but there was a kind of an equivalent forest type called the boreotropical forest even earlier than that in the Eocene. But in any case, the idea of widespread species distributions around the Northern Hemisphere. And then the idea that subsequent to that, there were extinctions, sort of differential extinctions. So we had more extinctions in Western North America as the climate dried out there. We had extinctions in Europe, especially with glaciation and so forth. So in the end, you end up with these Eastern Asian, Eastern North American disjunct patterns. So that's the basic idea. Now, the big breakthrough in this, in my view, started in the late 1990s. And the first paper along these lines, I think, that approached this in the way that we're approaching it now was by Jenny Zhang of North Carolina State University. Unfortunately, Jenny couldn't be here at the meeting. We invited her to our symposium, but she couldn't be here. But Jenny's a real hero in this area because she and Doug and Pam Soltis in 1998 started to ask the question, let's look at multiple groups of plants. There weren't too many of them, the dogwoods, boykinia, the ones that you see here, and let's see if there's a general pattern in the area cladogram. And they said, yeah, there is, actually. Eastern North America and Western North America are more closely related to each other than they are to Asia. That's maybe not a very surprising or thrilling result, but the idea of going and looking for congruent patterns was really cool, and these are the first guys to bring that to bear on this problem. At about the same time, Jun Wen, who I think is at the meeting, Jun Wen is another superhero in this whole area of Northern Hemisphere biogeography. She also looked at phylogenies, looked for congruent patterns, and produced a really nice paper that had a big, huge table of all these cases of Eastern North America, East Asia disjunctions. That's been super helpful. I got involved in this in 2001 when we published a paper where we said, let's look for general patterns, but let's see if maybe we can divide them into categories. So we said maybe there's an Atlantic pattern shown here in the phylogenies there, and maybe a Pacific pattern. Can we look at individual phylogenies of different groups and sort them into those two patterns, and how does that work out? And sure enough, we did find groups that had one or the other of those general patterns. But even with those few examples, we got onto the idea that there's problems, and the main problem is what's called pseudocongruence. So you can have the same phylogeny, but happening at different times and presumably affected by different factors. So on this phylogeny here, let me see if I can, yeah, I can sort of do that. This says congruence. And the idea is you have the same phylogeny of these areas, which are China, Japan, and North America, and at the same time, and those are about 10 million years ago. So that's a case of congruence. But a case of incongruence is the one that's shown here in this box, because here you have the same phylogeny, but at very different times. So one's at 5 million years and the other's at 20 million years or something like that. So this is the same phylogeny of these areas, but produced at different times and presumably by different underlying factors, right? So that's the idea of pseudocongruence. So what you start to develop is more of a picture like what you see over here, which is the idea that maybe things have moved around the Northern Hemisphere in different directions and at different times, right? So it's not all at the same time, and it's not all in the same direction. So then we expanded this with Stephen Smith, who was then a student at the time, now at the University of Michigan, and we said, let's look at these 4 major areas, let's go to the literature and compile all the studies that have been done and see what it says. So we did that, and I won't go into the details, but we displayed it in this kind of diagram where there was a Eastern Asia, Eastern North America, and the connection between them means that there was a disjunction in one of the phylogenies. And we find 50 of those different disjunctions out of 100 that we looked at the time. So half of them involved East Asia, Eastern North America disjunctions. And, but there were very few in other categories, like very few Eastern Asia, Western North America at that time. The other thing we could do is we could look at, over here on the right, we could try to decompose this and say, well, of all those 50 disjunctions between East Asia and Eastern North America, which ones was there movement inferred from East Asia to Eastern North America? Well, 20 of them. Which ones in the other direction? Just 1. And how many are ambiguous? 29. So that's how we summarize the data. Does that make sense? Now, the other thing we did at that time was we looked at age, but at that time people weren't doing dated phylogenies very well. But we did find 33 disjunctions with dates, and we categorized them simply as more than 30 million years old or less than 30 million years old. And for the ones that are less, we thought maybe the best explanation would be a connection through Beringia. This is a map in the late Oligocene showing a strong connection, whereas the North Atlantic was more discombobulated at that time. So the conclusion of this paper basically was this. It says many temperate forest plant groups originated and diversified within East Asia, followed by movement out of Asia at different times, but mostly pretty recently over the last 30 million years. So this came to be known in a sense as the out of Asia hypothesis. So subsequent to that, there were several other studies that took a similar approach, where they looked at the literature and tried to sort of say what was going on with it. And this was led by Jun Wen. You can see I've listed these 3 major papers, and Steffi Ickert-Bond. And they did one paper on Madrian-Tethian relations. That's the ones between Europe and Western North America. They did one on East Asia and North America, and they did one on East Asia and Western North America. And these were really helpful papers. And they summarized data in these kind of pie chart-looking things. And this is just one example of that. Trying to determine what was the likely migration route, using sort of the logic I used a minute ago. How many movements from the old world to the new world and vice versa, and so on. So anyway, we decided to do this now in 2024. And so with Erica Edwards, we started a project looking at the literature. And we're not done with this yet, but I'm going to show you the results as it stands right now. So we had inclusion criteria for studies. One, that it had to have a dated tree of a vascular plant clade. So we're not talking about bryophytes and things like that here. It had to have relevant disjunctions. It had to have a formal biogeographic analysis. It had to have dates for these disjunctions, which we actually put into big bins of 10 million years. We said, is it in the 0 to 10 million year category, 10 to 20, and so forth. And then we tried to say, what's the direction of movement? Can we infer that off the tree in the paper, and so forth? So that's what we did. I'll just show you what that looks like in practice. Here's a phylogeny of Acer and the maples. And you can see in this one, there's a lot of different disjunctions. Each one of those circles represents a disjunction that could be dated, and we could determine something about the directionality of the change. So just as one example, look at this one over here. That's a disjunction between Europe and Eastern North America. It happened about 25 million years ago. And it was a case of Europe moving to Eastern North America. So that's what we did. So we went through the literature and we looked at these papers and we assessed all of this kind of stuff. Now, sometimes it's complicated because people don't always do the same things. So for example, here's a study where instead of breaking North America into Eastern and Western North America, it's just lumped as North America. So we didn't really try to disentangle that very much. But we took it on board as basically a combination of Eastern North America and Western North America. Also, there are cases in these trees, many cases, where you can't really determine with much confidence what the direction of movement was. So we scored those as ambiguous. A good example would be this one right here. So this is a disjunction where you don't really know which direction it went. So we scored those as ambiguous, and we were quite liberal about that. So this is what a little bit of the data set looks like. And we looked at lots of papers, and in the end, what I'm summarizing here are 402 different disjunctions from these different papers. There are mostly angiosperms, but there were conifers and ferns and lycopods. We also scored habit, whether they were herbaceous or woody and so forth, and also dispersal, whether they were dry fruits or fleshy fruits, and tried to infer the directionality out of these things. So I'll summarize it sort of to begin with in the same way we did with Stephen Smith. So with these 4 major areas, there's these 6 possible disjunctions, and some of those are within continents. So Eurasia here is Eastern Asia and Europe, and North America includes Western North America. So those are intracontinental ones, within continent disjunctions. And then some of them are between continents. And of the between continent ones, you could be between continents but in adjacent areas. So for example, East Asia is adjacent geographically to Western North America. Does that make sense? But the other alternative shown in the middle here are these intercontinental disjunct distributions. So they're not adjacent on the map or anything. East Asia and Eastern North America is one of those. So those are separated on the map. It's a disjunction involving 2 different continents that are widely separated from one another. So the result from the Smith et al. Paper is shown. The Smith paper is shown on the left, and then the new results are shown on the right. And you can see that one thing that's happened is we've added a lot, right? So in other words, whereas we just had 1 East Asia, Western North America connection, we now have 50. And we had 19 East Asia, Europe, now it's 76 and so forth. So all the numbers have gone up fairly dramatically. But the overall picture hasn't changed that much in the sense that you can see there's still a very strong signal for this East Asia, Eastern North American pattern disjunction. So what about in time? If we put those in time, what goes on there? Well, each of these disjunction types occurs at all the different time periods. So there's got to be a lot of pseudo-congruence going on here, is the message there. So if you take this lower one here is East Asia, Eastern North America, you can see it's present at 10 million years, 10 million years, 30 million years, 40 million. It's present in all those different time periods. So that disjunction is coming about multiple times at different time periods. The other thing to notice is it's almost all recent disjunctions in the last 10 or 20 million years. Hardly any of it goes back to the Eocene or back to the Cretaceous or anything like that. So we have to change our kind of worldview here. Most of this is sort of modern stuff, actually. We're not getting a lot of signal from the deep past here, basically. We can sort of examine this from different angles. Like we could say how many are intercontinental versus within continent. There's actually more between continents than within in our data set. Those are sort of spread through time, as you can see here. Interestingly, there's more disjunct intercontinental distributions than there are adjacent ones. And of those, the predominant one is East Asia, Eastern North America. So this is the kind of picture we can develop. What about directionality? The directionality is very much like what we found before. Almost all of there's a very strong signal for movement out of Asia. And so I've circled some of those numbers there. So there's a significant asymmetry. Asia acts as kind of a source area for the Northern Hemisphere, but not as a sink so much. And you can see that actually in this kind of pie diagram. So as a source area, Eastern Asia is having a really big effect, right? That's the major source area for the Northern Hemisphere. But it's fairly average when it comes to a sink area. So the number of things coming into Asia from elsewhere is about the same as the number going into anywhere else, basically. And then I want to say that East Asia as a source has been a continual thing. You know, it's not just in the recent time or in the ancient time. It's in every time period, Eastern Asia is sort of acting as a source area when we can infer the movement. And the reason for that, by the way, that's been posited in the literature is that maybe East Asia is a place where lots of temperate groups have originated from tropical groups. And once they've adapted to cold climates, they can then move around the Northern Hemisphere through Beringia and so forth. That's what's in this diagram right here. So I just want to summarize this and then go to the last part of the talk. So there's strong support. There's lots of East Asia, Eastern North America disjunctions. There's strong support for a kind of out of Asia pattern. Most of these disjunctions are recent, and I think that's really fascinating because now that's right back to Philip Darlington. That's why I wanted to show you that. So he was much maligned for interpreting biogeography in a modern static landscape, but maybe he had a reason to do that because all these disjunctions are actually pretty recent. And actually, if you look at his diagram, it's pretty much the same as the one we developed, right? And a lot of groups originated in tropical Asia, moved into temperate Asia, and then around into other parts of North America, and then down into South America. This is just exactly what we're finding in plants, and on a recent time frame. Is there a single biological area cladogram? That's the question that Nelson and Platnick wanted to ask, and I think we can answer it definitively. No, the answer is no, there isn't any such thing. There's all these different patterns happening all through different time periods, and it's a very confusing mess, and it's not summarizable in any single area cladogram for the Northern Hemisphere. Now, what's wrong with this picture? There could be two things really deeply wrong. One, all the ages could be off. What if everything is systematically too young when we do our dating? That's a possibility. We can't dismiss that entirely. But you know, if you take this at face value, a lot of this stuff is fairly young. That's what we're saying. The other thing is that differential extinction in different parts of the Northern Hemisphere could actually cause a problem, an artifact. We could interpret something as coming out of East Asia when in fact that's the place that's just retained the most things that haven't gone extinct. And you have to sort of think that through a little bit. But that's a problem that really needs to be addressed by some clever people with simulations to see under what circumstances would that really lead you astray. But if you take it at face value, the results are what they are here. Okay, final part of the talk. I want to say, look, we've been looking at papers in the literature and started to summarize them. Each one of these is a paper that does an ancestral reconstruction. And the question is, are we doing a good job with those ancestral reconstructions? Because if we're not, then the whole exercise is kind of useless in a way, right? So we want to be doing the best possible job we can to do these reconstructions. And I'm going to use viburnum as an example of the work we've done to try to get better and better at doing ancestral reconstructions. So my first attempt at this was in 2005. And this was a good exercise, but it was small. It was 41 of 163 species. It had only one tropical species in it. It was just little snippets of DNA. We used DIVA, a parsimony method, and the trees weren't dated. So there were a lot of problems with this analysis when we look at it from a modern standpoint. So since then, we've now got all 163 species. We've got every one of the tropical species in the analysis, including these 3 from Borneo that are really interesting. And we've looked at all the fossils, and actually looked at them personally, not just in the literature. And there are a lot of viburnum leaf fossils that we've had to kind of just ignore because they're probably not viburnums at all. And Steve Manchester made this point in a series of papers, reassigning viburnums to Celtis and Davidia and other things. But I do want to make a note. I think there is an actual viburnum in the Paleocene, and it's this fossil right here, and I think it is actually a viburnum. But that's a whole other story. But in general, we've ignored the fossil leaves because they're pretty hard to interpret, and instead focused on fossil pollen in our analyses. And you can see a pollen grain in the upper part of this is from 48 million years ago from British Columbia. It looks very much like a species from Borneo nowadays. Anyway, so this is our biogeographic analysis from a paper with Michael Landis a couple years ago. And just relative to the East Asia, Eastern North America problem, what we find here is multiple cases, right? And we're finding pseudocongruence, right? So there were 2 that are here at the bottom, where there was a movement into North America from Asia at a fairly early stage, 55 million years ago. Here's another one maybe 30, 40 million years ago, right? But then these other ones are much more recent. So we've got 6 East Asia, Eastern North America disjunctions. 2 of them are older, 4 of them are younger. So that's where we stand on that. Now I want to talk about what was the first forest type? What was the biome or whatever where viburnum originated? What was it initially diversifying? Here there's conflicting studies. So we did a study that said it was probably tropical. This was the study by Spriggs et al. And we sort of tried to focus on the bottom of viburnum and say, was it in a tropical forest or a temperate forest? And then there was a paper in the next year by Lenz et al. That actually came to the opposite conclusion, using pretty much the same data set that we had gathered and so forth, but slight differences. And they came to the conclusion that it was most likely cold forests. So what's the answer to this? So this has been puzzling. But we've dug into this in a variety of ways, which I'm going to show you, and I think we can more definitively answer this question. So here's a biome reconstruction for viburnum, and if you look at the very base of it, it reconstructs viburnum as being in warm forests, not cold forests, okay? So that's the first conclusion of this. But I want to mention, whoops, sorry. I want to go back here. I want to mention a couple of interesting things. One is when viburnum moves in 55 million years ago into the New World, it probably did it in the context of warm forests, over here and over here. In these more recent movements into North America, the plants have already been adapted to cold climates in Asia, and they've just moved in and occupied North America, right? So that's how that's worked. I also want to emphasize that this analysis included fossils, right? And in that process, the fossils actually played a pretty important role because we assigned each fossil, we gave it a biome designation based on its associated plants where it was collected, right? So it turns out that the fossils had a pretty big effect on this estimation. So we weren't entirely confident about that. So with Michael Landis, again, we developed another approach. Actually, Michael developed it, but we used it on viburnum. And the idea here is to look at what's the distribution of different biomes through time. Can we take that information into account? So this is going from the Cretaceous, K there, up to the present, and at each time, each of these matrices shows the distribution of a particular forest type that we estimate around the planet, okay? And the most important thing for this analysis is that probably in the early parts of this thing, the early time periods, Eocene and so forth, there weren't very many cold forests around. That's the observation, right? So if you now take that into account in doing the biome estimation, the ancestral biome estimation, what you find is very strong support for viburnum originating in warm forests again. So this is consilience in a way. We're getting, from different approaches, we're getting the same result. Viburnum originated in warm forests and moved later into cold ones. But now the thing I want to show you, and this will take a minute, and this is the last little thing I'm going to do, is a new method that is being developed by a graduate student named Sean McHugh. with Michael Landis. He's a student of Michael Landis at Washington University. And he's developed a pretty nifty approach, I think, that we've just now applied to viburnum, and I'll show you the result that we get. But basically, this is saying, when we see a particular species living in a particular forest, we score it like, okay, yeah, that's living in a temperate forest, or that's living in a tropical forest. But we don't know, could it actually live in another forest? We don't have that information, right? So the realized niche might be different from the fundamental niche that might include some more forest types, right? So that's what's shown in the upper panel here. We have the actual true species biome affinity repertoire, but the only thing we see is the green one there, and we don't have any information about the fundamental niche or whether it could live in another type of forest. But the question is, could we bring information to bear on this? Could we actually bring information to bear? So we have two sources of information that we're using. One is botanical garden records of whether plants can actually grow in that botanical garden in a climate outside of where the thing actually grows in nature. So that's a form of information that we can take on board here. The second form of information is about, in this case, about germination requirements. And I'm just going to show you why that's important in viburnum in the next slide. So these are bits of information that we can add into the inference problem and make even a better estimate of the ancestral area. This germination thing is interesting. So it turns out that some viburnums that live at northern latitudes in cold climates, they have what we used to call double dormancy. It's now called deep, simple, epicotyl morphophysiological dormancy. But basically what it amounts to is these plants have a tiny little embryo, and they have to grow for a long time to get through the endosperm and finally put out a little radical. And they do that after a long period of warmth. But then they need a cold period of like several months of cold before then they'll send up the shoot. So they send down a root and they sit there, and then later, after cold, they send up the shoot. And that, I won't go into the details of this complicated diagram, but that's what that shows at the bottom. So we can show that some viburnums have this double dormancy. Now, the significance of that here is that if you have that, if you require a cold to germinate, it's going to be hard to occupy a tropical forest where there's no freezing. Do you get the reasoning? Yeah, so that's the kind of information we're trying to take on board here. So this is the kind of result that's produced by this new method. It's called the RFBS model, which stands for Realized Fundamental Biome Shift. And at the basal node of viburnum, I've sort of blown it up over here. That's what the basal node of viburnum looks like. And it's reporting 3 bits of information about the likelihood of occupying a cold forest, a warm forest, or a tropical forest. And what we reconstruct in this analysis is congruent with everything I just showed you. Viburnum is likely to have originated in a warm forest. So this is the realized niche and also the affinity probability. I can't go into the details of the method because it's a bit complicated. You just have to read the paper. But basically, you get the idea of what we're trying to do here. But I want to show you one case where it makes a difference, where it's changed my mind about something. So this is Porphyrotinus. This is the clade that enters North America 55 million years ago. We had reconstructed it as being in warm forests, and that's confirmed by this new analysis that takes on board the germination requirements. So that nothing changed there. But here's a case where we had reconstructed for this particular small clade a likely warm ancestor, but now in the new analysis that takes on board the germination, it actually is showing a more likely cold ancestor at that node. So it's actually changing something about the way we're inferring this, right? And most importantly, from our point of view, is we've been studying a lot the ones that moved into the cloud forests in the neotropics. But we've always been unsure, like where did those come from? Did they come out of cold habitats from the north and occupy the cloud forest? Or did they come out of other warmer forests around America and then occupy the cloud forest? Well, it was very obscure and equivocal in the analysis we did initially. But with these constraints added in about the germination requirements, it now becomes pretty clear that these occupied cloud forests coming out of warm forests, not coming out of the eastern North America, the way I always used to think about it. I thought they were cold adapted, then they had to move into the cloud forest and then lose all these cold adaptations. Now I don't think that, based on this analysis. So the point of this is we can take on board a lot of different kinds of information. I just showed you we can take on board fossils and their environments. We can take on board the distribution of biomes in the past. We can now take on board things like germination requirements and these botanical garden records, and we can get better and better inferences of these ancestral areas and ancestral biomes. And I think that's where we're going to need to go, is take into account more information, make each one of our inferences better, and then then these general studies are going to be that much better, basically. So to conclude, I showed you a little bit about the latitudinal diversity gradient. We talked quite a bit about Northern Hemisphere biogeography, and then this point at the end about let's make our inferences as good as we can, and let's try to be clever about how we bring in as much information as we can. And with that, I think I'm out of time, but I just want to say I'm so grateful, so happy to be here. So muchas gracias to everyone, and especially the organizers, and especially I want a special shout out to Gonzalo Nieto Felanir, who has taken the lead on this meeting and done such a wonderful job with the organizing committee and everybody. It's just a fabulous thing that you've put together, and it's a delight to be here with you in Madrid. Thanks also to my colleagues and funding sources. Gracias.
Thank you, Michael. Really cool science. We have around 3 minutes for questions.
I think I'm over here, Michael. Here. Can you see me? This way.
Where are you? Oh, you're over there.
So thank you for a wonderful plenary talk. I learned a lot, and I have a question sort of coming from plant ecology. So I was wondering how you think that functional attributes such as plant pathogen interactions might be useful in generating these reconstructions. And I'm just going to cite a really quick example of the chestnut. It's what inspired me through this talk, that the chestnuts in Eurasia are resistant to the chestnut blight, and the American equivalent is not. And it seems to me that that is strong, or at least supportive evidence for the fact that that may be that the trees moved, but the fungus did not come with them. So I was wondering if you had any insight as to how that might be useful in using these in generating these reconstructions.
Yeah, see, what you just said is so interesting to me. I think everybody heard what you said, I hope. That's another kind of information that in the back of our heads we've been thinking you could take on board in these reconstructions, is the relationships to different, and it could be pathogenic relationships, or it could be mutualistic relationships, it could be any kind of relationship. But taking that on board and making these ancestral reconstructions is honestly something people haven't thought about doing. And we don't at this point have a model to do it. But I think it's a really great idea, and I would love to pursue that. In viburnum, by the way, we do know a lot about that. And there is this thing called the viburnum leaf beetle from Asia that's eating all the viburnums in the New World. And that's it's a case very similar in a way to your chestnut. And I think we could probably take that on board too in making some assessments. I love that idea. Thank you.
Before we move to another question, just to say that Gonzalo has an important information to announce, so I asked people not to leave the room until this finishes, just 2 minutes. So one more question.
Thank you for the fascinating talk. I enjoyed all of it. I have 2 joint questions. So one, have you looked into the Gondwana and then more recently the South American origins and then the subsequent disjunction events? And then in relation...
Wait, say that one again, looked into the...
Have you looked into the Gondwana origins of species and then, or more recently, South American origins and then their subsequent distribution events? Because I noticed in the Laurasian disjunctions, everything is more recent in time. So if you look at more ancient clades, are you seeing anything there? And then the second shorter question is, when you talked about colonization events, how do you suggest we look into that information? Because some of these disjunction events are very old and we have limited information on how long a species has been in that area. So do you have any ideas on how we could investigate that?
Yeah, on the first question, the whole Gondwanan biogeography situation, the southern hemisphere is a different story altogether, which obviously I didn't touch on. But this is something that Isabel and many other people have looked at southern hemisphere biogeographic patterns. And there it's much more, it's a little more interesting in terms of general area cladograms and how it matches up with the breakup of continents. And there are a number of old southern hemisphere lineages that probably are affected by continental drift in that way, but many of the northern hemisphere ones, as I've shown, are probably not that much affected by continental movements. So the southern hemisphere is very interesting and kind of a different story altogether, which would be fun to also do this kind of meta-analysis on that. Yeah, the second one, I mean, about how exactly things got around and colonized different areas, especially if it's deep in the past, that's tricky. If you take my message seriously, one bit of good news is a lot of these are fairly recent. They're not, it's not going back to the Cretaceous and where we have very little information. A lot of it's probably within the last 10 million years at least. So. All right, thank you again, everybody.
Thank you.